Okay, so it's my pleasure to introduce our final speaker. Now, Julien was so Julien came in quite a short time ago, so it was a bit difficult to arrange travel for Julien, but he's doing such great work that I think it's great to have him here virtually anyway. He's been doing great work on language models recently and lots of cutting-edge deep learning methods generally in the last few years, and really focusing on getting these methods to work in real situations, you know. So I think he's really sort of walking the walk, not just talking the talk. But today he's going to talk the talk because he's going to give us a talk. So over to you, Julien.
Thank you. Hi, guys. So, yeah, thanks for the invitation. I understand you guys have had like 2 days of very packed schedule with a lot of information. So I felt I'll yeah, have a presentation for an hour and a half, but probably it's better if you guys just interrupt, we discuss, and we go to some extent through the slides. It doesn't have to go through everything. I will also put a little bit of, you know, fun videos, not too many equations, pictures, to make it perhaps easier to grasp at this stage of the summer school. So let me start with this, just as a motivation. You can see the screen?
Yeah.
Great. Okay. So what you see here is an embryo of Drosophila, and you're seeing nuclei, perhaps already cells. And these are the very first, you see the time up here, the very first minutes of this developing embryo. And a lot is happening. Cells are finding their ways. You see a lot of divisions. Perhaps you can guess that all these cells start to have different stages, different types. Structures are coming up. Those are very fast, right? And coordinated. These instructions are remarkable. You also have to see that at least throughout this entire movie, every cell has exactly the same genome. All the cells many cells are dividing, and they're copying the entire genome, and it's always the same set of instructions. And these things still in a very coordinated fashion are happening. And that's true for the fly and for all species out there. I guess I can stop here and perhaps just fancying things like this. Try to still share screen, sorry. Is it's remarkable. And so my talk today is about how we can get more insights into what in the genomes, you know, where are all those instructions, and how can we use techniques from machine learning or artificial intelligence, or however you want to call it, to help us deciphering this. So what I just told you is that the information in the genome that you have in the very first fertilized egg cell contains all the instructions for the entire development, the entire body plan of an organism throughout all the stages of life. It has also variations in the genome underpin much of the phenotypic variations we see within species between individuals. And there's unfortunately also errors, if you wish, or variations again in the genome that underpin inherited disorders, genetic diseases. And it is mostly acquired mutations still in the genomes of cells that is the hallmark and one of the major causes of cancer. And therefore, having better tools to interpret genomes helps biologists to better understand the mechanisms underlying life. It better helps us understanding how individuals differ, and some are more at risk for particular diseases, and understand cancer. It's also, I think, important to realize for all of you who are working in health, in the context of health. Yeah, you guys are working with data. You have your favorite data modalities. You have your favorite types of models. Whatever you do, I want you to really keep in mind that we have with the genetic information something that is absolutely unique and very important in the fact that it has a very rich information regarding causality. Yeah, and the reason is that the genetic, the inherited information that you have in this very first cell came first before anything else that happens afterward. So temporally it comes first, and therefore you mostly have a flow of causality that comes from genotype to phenotype. And that is extremely valuable when it comes to causal inference, to understand causal relationships between variables that we see in biology and in medicine. And as a matter of fact, most of the drugs that are successful have actually a genetic evidence. And also, we can see during the different clinical test phases of testing of drugs that the most successful ones are those that are supported by a genetic evidence, where we have from the genetic evidence that the underlying the target is actually causally predisposing to disorders. Okay, so it's something that I would say whatever we do, however, which type of data modality you're working with, I think having a piece of genomic information behind or a connection to it can be extremely helpful. Okay, so the outline of my code today, talk today is such. I want, in the very first part of this talk, give you just biological background. I'm not sure what is your background, and so it might be helpful that give you a bit of definitions and understanding of what we mean by the regulatory code. And then we'll go into stages. One that is essentially a review of what has been done in the last 5 years or a bit more in modeling the regulatory code. And all this has been essentially based on supervised modeling approaches. And then more current research. Not sure we'll have the time to get there, but there's a lot of work nowadays on self-supervised modeling, and I can perhaps have the time to show some of our recent work and even unpublished work on this front. So far, so good? Yeah, yell if there's anything, and hopefully I can hear you. Okay. So first of all, how do cells read the genetic information that is in genomes? That is what is called gene expression. So we have in the human genomes protein coding genes. It's about 20,000 of those. You can think of them as intervals in the genome. And these are in the first step transcribed. So RNAs are produced out of DNA. Multiple copies of this interval can be made. These are called precursor RNAs. These RNAs themselves are processed. One of the major processing steps is called splicing, in which introns, entire intervals within the RNAs, are removed, and the remaining ones, called exons, are bound together or spliced together. My picture shows only one intron, but a typical human gene will have about 10 introns. These processed RNA then get exported outside of the nucleus. It's called, in the case of a protein coding gene, it's called a messenger RNA or mRNA. And this messenger RNA has a message, that is the coding sequence. And that is the basis then for the translation machinery to produce now an amino acid polymer, a protein, that is the actual molecule that does much of the functional work that this gene is encoding. There are more genes, also non-coding RNAs, but understanding this basic principle here is good for today. Okay, so this flow of information here is called the central dogma of molecular biology. Information is encoded in DNA and then transcribed into RNAs and translated into proteins. There can be feedback here at this level, but it's not back to DNA. So far, so good? Great, thank you. Right, so now how does this function? How do our cells read this? What instructs cells to start transcribing a gene? I will start by explaining very basically what happens on transcription at the transcription level. We have in cells proteins called transcription factors that have the capacity to bind to DNA, and they do so in a sequence-specific fashion. So they are in DNA's so-called transcription factor binding sites, which you can think of as words like this. And that molecule, that protein here, has a particular affinity for that word, such that there can be binding with more or less frequencies, with more or less shorter or longer durations. Upon binding, a transcription factor has the capacity to recruit to the DNA the transcription machinery, so-called RNA polymerase, that will then grab a particular position in the DNA and open the DNA and then start to make a copy of that gene. So you have synthesis here of an RNA all the way to a particular place where the transcription will be then stopped. Okay. Now, so this can happen in several rounds. So if this event is frequent, you may have multiple rounds of transcription or multiple transcription bursts, and therefore a certain number of copies of RNAs that are made with although the DNA remains is in a single copy here. Okay. So the implications of what I told you, right? This little word here, that so-called regulatory element, plays different roles, or is important for different aspects of gene expression. First of all, that location in the genome where this is found is one of the components that determines where the transcription will start. And therefore, it contributes to definitions of genes, at least of gene starts, right? So it tells you what is transcribed, what is read in the genome. The second point is the quality of this word. Here I just wrote TCGTA. Okay, that might be for my, let's say, thought experiment, the words that transcription factor likes very much. So there's a very strong binding, but maybe if there's a change here, that the frequency of those events is smaller. Yeah, so the quality of these words has a quantitative effect. It determines the frequency of this event and therefore of the transcription. And then last but not least, these proteins here, these transcription factors, they are themselves encoded in the genome. They are themselves under the influence of other regulators, especially whose activity depends on the timing, as I showed you during the embryo genesis, or of external stimuli, of stress, etc. And therefore, how much you will transcribe from a gene depends on when and where that cell is, as well as under which condition. So it also has instructions about what to express at what time point. Okay, so far so good? Yes. Great. Okay. There can be variations in the genome that we see in patients, and these that we see across individuals, and some of them can lead to disease. You can imagine that a mutation on a transcription factor binding sites will alter the quality of this, and therefore the frequency of bindings, and reduce the expression of a gene. We see also sometimes in cancer the opposite, that a mutation create a binding site that usually is not there, and therefore activate a gene in situations where it should not be active. For example, triggering proliferations of cells in states where there should not be proliferation happening. So genetic variants can, by disrupting regulatory elements, contribute to diseases. Okay, So far I explained you everything very conceptually for a single transcription factor on a single transcription factor binding site. But what you have to see is there are thousands of, more than 1,000 transcription factor are understood to exist in the human genome. And those can have different affinities for DNA. So there's an entire lexicon of motifs that contributes to this regulatory code of transcription. Moreover, the spacing, the orientation that can have an effect on this transcriptional output. And so there's not just a lexicon, there's even an entire grammar that dictates how good transcription perform and in which condition. Not only this, but what I told you for transcription is true at every steps of gene expression conceptually. And those will be different words, there will be different factors, but you also have within an RNA element that can be recognized by other factors. For the case of splicing, they're called splicing factors, and they also have affinities for particular sequences, and therefore would lead to splicing at more or less high quality or in different conditions. The same happens for translation. There are motifs which are present in this part of the RNA that is not coding for the protein in the so-called untranslated regions that contains motifs which will modulate how effective translation happens, and therefore how much proteins you have per mRNA molecules. Also, there are motifs that are involved in translation that are found in the other untranslated region, the 3' untranslated region. It's not all about production, it's also about degradation. This matters a lot to define the abundance. Abundance of a molecule is the balance between its synthesis and its degradation. Moreover, degradation is important for stress, for temporal responses. An RNA that is quickly degraded its abundance can be more rapidly altered by between conditions. Something that is very stable, in contrast, will be buffering variations in transcription. So this is also a place where regulation happens, and there's also an entire regulatory code which instructs cells on how much degradation of RNAs, as well as on proteins, with different factors and different code happen. So far, so good?
Yeah.
Okay. So altogether, these are at each stage, you have thousands of possible factors, you have hundreds of kind of motifs, you have entire grammar, and this regulatory code is not deciphered. As of today, people have done, the community has done quite a lot of progress on the transcription in mammalians, thanks to projects like ENCODE and a lot of modeling done on these data. Post-transcriptional code is much less studied. And other species are also less studied. Okay. Okay, I don't know if I show you that movie. So if everything is fine, I can just move on. So this is a nice movie you can look at if you want to see splicing happening in an animation, right? And that I find it fascinating because when you see videos like this, and you become aware of what happens in your cells at any point of time in multiple molecules in parallel. This is just mind-blowing. And the importance, I think, of splicing, that's why I like showing this video and also in my teaching, is it looks very chaotic and very complex for cells to manage this, but you have to see that the exact base has to be spliced at the right position. And if this happens just one base, by a shift of one base, it completely disrupts here the coding sequence, because you get a non-multiple of 3 shift in the coding sequence that can completely kill the protein. So those things that looks chaotic and very complex are under very strong selective constraints to happen relatively precisely. Okay, so far so good. Yeah. Okay. I just don't hear feedback. So I mean, I heard so far when you said. Okay. So how do we crack that? So there are three major activities of the community, the scientific community, to crack that code. On the one hand, people are developing and applying assays which allow us to probe all those steps of gene expression genome-wide. And now also many of these assays exist at the single cell resolution. So those assays leverage, many of them, right, leverage high throughput sequencing, because you can turn basically the measurement of those things into either sequencing DNA or RNA. And then there is, for the protein level, also mass spectrometry-based assays that allows us to quantify, even to sequence to some extent, the proteins. Also now going down to smaller and smaller amounts. Even some first single cell mass spectrometry assays are available. Okay, so that means we can know and measure, let's say, where transcription factors occur across the genome. The ChIP-seq protocol is one such protocol which allows you with an antibody to identify where a transcription factor is bound in a population of cells. Cells and genomes are fragmented while still keeping the link between the transcription factor and the piece of DNA it is bound to. And those pieces of DNA are then released and amplified. And so in the sequencer, you can then count how many fragments you identify for a particular position. So you get signal that looks a bit like this, like bumps across the genomes. That can be quantified and then be the basis for modeling. And this concept here has been deployed at other levels. So if you look at where RNA binding proteins, which are those guys that are important for the life cycle of RNAs, its synthesis, its translation, its degradation, its localization. Those guys also can be done assayed similarly with another type of assay called CLIP that also tells you quantitatively, statistically, where do we statistically find specific RNA binding proteins on RNAs. An important assay is RNA sequencing. I guess I'm not sure whom I'm talking to, but I guess you guys have heard about, in which by sequencing RNAs, we can quantify how much of an RNA is in a particular population of cells or even at single cell resolution. And then we may also, especially with assays that are in cell populations, quantify the abundance within a given transcript of particular regions. So therefore, one can estimate how much of the mature RNA is over the precursor RNA by looking at the ratios of reads that come in the exons, which are found both in the mature and the precursor RNA, over reads that are found only in the introns, which are primarily stemming from the precursor RNA. And we can also see quantifications of different exon usage, and so go into exon choices. Okay, same on translation. There are assays that allow you to see where the proteins involved in translation are present. Again, CLIP for the RNA binding proteins. And there's a protocol very important for studying translation called RiboSeq that is able to identify the reads that are at a given that are protected by a ribosome. A ribosome is the translation machinery that creates the proteins from the messenger RNA. And so one can quantify for every RNA how much ribosomes are found per mRNA molecule, which is a good indicator of how much translation is occurring. But we can also see across the RNA variations of ribosome occupancies, which may point to situations like ribosome stalling or acceleration or deceleration of the translation. So those things are extremely quantitative and can give you also ideas of quite in fine resolution while still being genome-wide about these different steps of gene expression. Okay, all good? Do you have questions on that? Shoot. Okay, I continue? Yes. Good. Okay. So this, to me, these are one of the major aspects of what we do in biology to crack this regulatory code. These are genome-wide observational data, right? You observe, you measure, you quantify different steps of gene expression. Another category I'm using here, a figure from a paper from Georg Selig lab, are our capacity as a community to synthesize artificial genes. And this can be done with ingenious fashion in very high throughput ways. Small pieces of DNA can be engineered at high throughput and also assayed in parallel. So in this paper here, we are looking at a so-called massively parallel reporter assays, which are genes where, in this case, you have a study of splicing. So this is maybe the easiest place to look at. We have the authors have been asking which pieces of which elements in the genome are important for splicing or not, or skipping a particular exon. And so what they've done is they engineered a gene that has elements which have a splicing event and which contains elements that were synthesized with random sequences. So you have to see that every sequence is by chance different than another one, and altogether it could generate over 2 million of those sequences. And then it could be assayed together, and thanks to the sequencing of these elements, the authors were able to quantify how much splicing happens with respect to identity of this sequence. All the rest is the same, all the rest is equal, so you get rid of a lot of confounders by having a design of this kind, and build models then that can really explore the sequence space in a much, much more systematic way than if you rely only on sequences you find in let's say, across the human genome. So this is for splicing, but there are many, many papers that look at variations of this concept where you can engineer genes and test, again, all rest equal, the effects of variants, and give you very powerful models that can generalize pretty well in unseen sequences. Okay, so that's the second thing. So we can measure very well. We can also design. We can read and we can write. Let's put it like this. And then the third part, this is, I guess, where you guys kick in. That's we have also, thanks to progresses in machine learning, extremely flexible classes of mathematical function that allows us to fit models to such data. And so there things have been kicking in 2015, early 16 in our field, with the first papers showing how deep learning methods can be very nicely applied to this type of task. We had the chance to write a review with the group of Fabian Theiss on this, and that is one of the main figures of our review, where we didactically, try to explain what's going on in those models. So here, what you see is an architecture of a standard convolutional neural networks. Convolutional neural networks are still extremely used in our field for modeling from sequence particular event. So in this cartoon that explains the principle, assume you want to predict the binding of 2 transcription factors to DNA. And in this cartoon, we name them GATA1 and TAL1 from the sequence. So what you have as input is your DNA sequence here on the left. Your goal is to predict this as maybe a binary or a quantitative value. Okay. And the first step is to transform this sequence here, which has only 4, every nucleotide has only 4 possible values, right? A, C, G, or T, into an image. Yeah, so one-hot encoding is this very simple trick where you map basically every nucleotide to one of these 4 values. So you switch on the pixel, if you wish, and this 4 times L, where L is the length of the DNA sequence you're considering, and keep to 0 the other pixels. So now I have an image, and once I have an image, I can do image processing, and convolutional neural network is the basic thing to do. The nice thing is there's a very nice relationship actually between this and how people have been modeling quite successfully in some situations, binding of DNA, of transcription factors to DNA, at least in test tubes on very small sequences. And what people have found on test tubes in very small sequences is that you can, with a very simple 1-layer perceptron, model maybe for 90% of the transcription, single transcription factor, every single DNA. They're binding affinity. And so a single layer with just one filter, it's long enough, will actually capture not too badly what a single of this transcription factor does in a test tube in front of a piece of DNA. And so the first layer of these convolutional neural networks were essentially recapping what we used to be able to do before. But the beauty here is that the field was a bit blocked before deep learning at moving up to the next layers, which are like the interactions between the transcription factors. We had to learn cutoff, engineer those cutoffs, define some kind of class of functions for interactions as a function of distance. And all those things were very tedious and not very performant. As soon as we moved to deep learning, we could have like end-to-end models that would really, from the sequence all the way to this output, will learn not only the filters, but also how to integrate and model interactions between the different motifs. So all this is trivial, perhaps today, but it has really been a revolution in the field of bioinformatics to, as in other fields, right, to stop to have feature engineering and have really models that can train everything end to end. So far, so good. I assume you're familiar with convolutional networks, so I don't have to explain this picture in more detail. But let me know if you have questions. Maybe it's a good time to ask first questions.
Any questions? We're all good, Julien. I think they want to hear the rest.
You want to hear the rest? Okay. But
Question.
people, I can't see pictures, I mean, faces. And are you guys like asleep because it's too much or?
Julien, can you hear okay?
What?
Can you hear?
I can hear, yes.
Okay, let me just.
I can also just shout. Can you hear me if I shout?
Yes.
Okay. All right. I mean, it's a really simple question. I'm not sure what you're predicting here. You have some transcription factors binding onto some sites. And are you trying to like predict the amount of some protein that gets produced?
Good question. Well, it's good to have a question. Yes. No, so sorry. In this cartoon, you mean?
Yeah.
Oh, it's only binding. So you have a piece of DNA and you know it's bound, not bound. There's no production, right? So the production, there's very little models that do that well. Like you start from here and you go all the way to protein abundance, driving, traveling all the layers of gene regulation. On transcription, there's some models, but that is not ready. The field is not ready for that. But what we model here is just this step. Is TF1 bound here, right? Very simple. Very first step. Just the binding of the transcription factor to DNA, not the final abundance of the gene. So far, so good? Does it answer your question?
Yeah, thanks.
Okay.
One more.
Yes.
Hi, Julien, thank you very much. I was just wondering if it's possible, if you think it's enough to just use the sequence to learn these interactions, or we need the gene expression also to include using some sort of model data. Because if we, for example, study developmental samples, I just wonder if the sequence is enough.
Yeah, so, okay, thanks. That's the general concern of this. So, what is good for me to show that? I don't know. Yeah. So these abundance here depends indeed on the condition, right? So how do you model that? If you have data for many conditions, basically what you do is you predict, you do multitask. That's the most simple way, right? Multitask, and you predict the abundance in different conditions. And so the state-of-the-art models, they do that. They have done, you capture a lot of measurements of different layers for many conditions. And each of these measurements and each of these conditions is a particular task. And then you start from DNA. So you condition on your environment and cell type. generalizing across cell type will mean that we have good ways of representing the cell type. And that is ongoing research.
What I meant was that sometimes what we observe is that the chromatins are open very early development stages. But from, for example, very early stages, they're open and they are still open for some time. So I'm just wondering if this will confuse these models. If I want to study the regulation at the later stages, but you know, the chromatins are open for a long time, then how these models are going to differentiate this. So I think that in these special cases, using also RNA expression is important because the chromatin is open, but the gene is not expressed. So it kind of helps us to remove some of these false positive agents. I just wonder what you think.
Yeah, so you say if there's some memory that on some sites the sequence may be the same, but so the concept here is that you want to connect what in the in the sequence, even very far, right? You can have a very large receptive field, is determinant in cis for the stage you see. And if these things, let's make a thought experiment. We can have, I mean, there's more than a thought experiment where we have many situations where you have two copies of the same piece of DNA. If that is enough to define your chromatin state, then you can still capture that. It is definitely not true for everything. We see on X chromosome, for example, there's just a random choice of which one is active and which one is not. And there's very little way to figure out which one will be, which one will not, just from the sequence of the X chromosome. But otherwise, the fact that the chromatin is open may not be because of elements that you see, of factors that you see at this time point, maybe for events that have occurred before, but it's still encoded by this piece of DNA in cis. So at the first hour of development, that piece of DNA had some instructions to become closed, and this is why it's still closed now later on. So the task of predicting the gene expression just from sequence is still valid. You see what I mean? So some models are using external information beyond DNA as input, and they run into extremely dangerous, I think, claims because you have some kind of, you have like non-causal features in your input, and you start to have collider effects and things like this. If you start to do a regression on things that are actually consequence of what you want to predict, that is, you may improve predictability, but you can have very weird then variant effect predictions and then causal interpretations of your model. So we and many others are stepping completely away from that, and we just do DNA based to keep the the causality in the causal let's say behavior of the model. Does that answer you?
Okay, we got one more question here.
I want to expand a little bit on the previous question. So between the genomic and transcriptomic layers, there is also the epigenomic, which can also work as a confounding factor, right? Wouldn't this, the prediction task that you're trying to do there, wouldn't it be helped also by accounting for, one example is accessibility, but there might be also methylation, or you might, because if I correctly remember, sometimes methylation helps in the transcription factor finding, but that might be totally wrong, right? Or
that is true. But the same applies again. Why is this thing methylated?
Sorry?
Why is something methylated? And the questions we have with sequence-based model is, can you predict from sequence why you're methylated? So methylation is, is also encoded in the genome eventually.
He doesn't know.
I don't know.
He doesn't look like he knows, Julien, so we're gonna let you get back on with it.
Okay. So I continue, or?
Yeah, continue.
No, but those are like the important and relevant questions, and often it's this. It's like you have other features that are important for gene expression than just the DNA alone at any time point, but many of these also have a genetic basis eventually. I mean, from your fertilized egg cells all the way to where you are. So even though the mechanism at a particular time point involve other things, those guys have a genetic basis, and what those models are aiming at is completely to predict, the entire thing just from sequence. So what is basically encoded in DNA that can predict your state? And that may involve things that you don't measure, but that are also influential, such as a methylation state also. All right. It's not that you can still have other things, but much of those so-called epigenetics eventually can be also or are influenced by genetic themselves. And there's little also transgenerational in human, for example, transgenerational passing of information outside genetic.
Genetics also capture like environmental factors, right?
So this is different, right? So the environmental factor, that's something you also condition on. You say, in this particular situation, what is the expression level of the gene? And that is part of the covariates of your model for these types of models. You're not predicting that in every cell type, the expression is the same. You say, for that cell type, given my data, can I predict gene expression? With the goal, like, would there be a SNP at this position? What would be the effect? Okay, that's a very philosophical discussion. But maybe we can move on. Yeah?
Okay, yeah.
Okay. So back here. All right. So what are the new trends in the field? So we have models that try to predict everything at the same time. All what we could measure directly from sequence. One of the famous preprints, still a preprint, from Linder and colleague is the model Borzoi. Another one that is famous, which is published, is Informer from Giga also colleagues also from DeepMind and Calico, and they predict a lot of measurements, including what we just discussed, DNA accessibility, methylation, RNA sequencing, chip sequencing, essentially everything you can get from ENCODE. And these are a lot of tracks. There is, for the Borzoi example, over 7,000 of these genome-wide tracks. If you look at all possible modalities and all possible cell types, the things have been measured, and over 2,500 in mouse. So that's a lot of data, which often have even a single base resolution. What Borzoi is able to do now is a 32 base pair resolution prediction from sequence with an input sequence that is alpha megabase. So that's massive. But it has pretty remarkable performance. So you get really the profile you get for many assays, quantitative accuracy that is in the as good as what is a replicate experiment. So that's pretty strong. Still, there's still some limitations of those models. It captures transcription, not much post-transcriptional regulation. Not for all the assays you have the prediction that is accurate. So transcriptome remains hard, especially parent effect of long-range effects such as enhancer to promoters are still not fully captured. But that field is really going into, I guess, eventually we'll get to the single base resolution, multitask from sequence. Another trend that we observe is using only genomes. So in these situations, we have people that use large language modeling that apply to DNA. So here you have so-called unlabeled data from a machine learning point of view. That means we have the input of our models, but not much the output. We trained from the input alone, just from the genome models. And the nice thing for this approach is that there are many, many genomes, actually. It's much cheaper to sequence one genome than to do all those assays that I mentioned to you. And you can do this for a lot of species. And so there are many projects that who aim collectively at essentially getting genomes of all species out there. And there's already a lot of eukaryotic genomes, like more than 10,000 that are assembled, even more that are in the process of being assembled. And those are very large. Each genome is pretty large, right? You get from the million from small genomes to 10 billion genetic genome size. So it's a lot of tokens, a lot of things you can use for training. And there are another reason why this is good. It's not just you have a lot of data, is the assumption that is behind self-supervised learning, that is the regularities you see in the input in X, the features that you can find there are also those features that are helpful for your predictions. Well, there's an argument why this is applicable to genomes, is that natural selection enriches genomes for functional features in specific context. So there is constantly mutations that will basically purify regions that are not useful and enrich for those that are functional, and this in the particular context. So applying large language models is one way to reconstruct those, to detect such features. And this is something that is quite widely happening at the moment. I'm showing you one from our research. What you see here at the top is on a human promoter for the LDL receptor, which is an important protein for lipoproteins in cells and in blood. And what we get here in this plot here is each nucleotide scaled by the probability that this transformer gives for a particular nucleotide given the sequence context. At the bottom is another track for the very same promoter, but that comes from a mutagenesis screen. So here, this promoter has been placed in front of a promoter. Sorry, I displayed it in a reporter assay, and the transcriptional output has been measured. Well, so wait a sec. Okay. Can you hear me again?
Yeah.
Yeah, okay, sorry. Good, so what you see at the bottom now is the effect as measured by a mutagenesis screen. So every base has been mutated, and then the transcriptional output of that promoter measured, and so the effect here is the effect of each nucleotide. So those that are important for the output. And there's qualitatively a pretty good correlations between the two. And that's quite remarkable because the top one is only unsupervised, right? It's just trained on with a transformer on pure genomic sequence without training like the models before. I showed you the border that has been trained on actual gene expression output. So this type of approach doesn't beat at this stage what you can do with so much labeled data, right? Like training on ENCODE, but it works remarkably well, and it could also be helpful for improving models that are trained in a supervised fashion by having a better representation of the input, but also to tackle other problems for which we don't have as much data as ENCODE, anything post-transcriptional, for example, or gene regulation in other species. Think of bacteria, for which we know much less. Think of many pathogens, fungi, plants, a lot of organisms that are of relevance, also medically. Yeah. Good. So to summarize a bit what I told you in this first part, regulatory elements are elements in genomes that are involved in the control and of gene expression. They can be probed by genome-wide assays and high-throughput perturbation assays. And we have extremely large labeled and unlabeled data that are amenable to deep learning techniques to model this. As of now, there are many regulatory elements that are known, but we are not sure how good or comprehensive we are. There are still things that we can't predict, and so we don't know how much of the regulatory code is missing. And then there's still many non-coding variants which we find associated with disease that probably are the cause of these disorders, but that remain non-interpreted because they are in areas where those models are failing. So it's still hard to interpret the genome of an individual and make very clear prediction as to what is happening in terms of gene regulation in any cell type. Okay, so do you have further questions on this first part? And then I go to the second. We're good?
One over there?
Just speak or
Yeah, go on. CP.
hi. So I really like the slide with the many genomes and
Okay.
in the DNA language model. And if I decide to train a model on all these genomes, how do I sample from all of these genomes, and how should we model the training set?
That is not clear. There are things that it's active field of research. What we have done is we have so some people have looked into training only across human genome, and that is not working. I would have guessed, but because the trick of this self-supervised learning here is really to tap into evolution. Yeah, and so you get way more information if you start to go throughout a species. We have trained a model. I could go to this. That's the last part of my talk, where we've done species-aware DNA language models, and there we focused on fungi as a kind of playground to establish these techniques. And so the model is leveraging a lot of species. The evolutionary span we look at is so large that actually the regulatory code is evolving. A bit like if you have, Old English and Modern English and German and so on, the several languages kind of. They're related, right? But you're trying to cross all this. So what we do is we give on a token for what is the species, and the model learns how to map species in an embedding that allows them to reconstruct the sequence. So it's adapted, and there are a lot of commonalities, and there's also some evolution that is captured. So my answer is you better sample faraway species. Don't say just within human, otherwise you just learn there's not enough variations. Now it will depend on what you want to do with it, and it's an area that is not searched enough. And it's not clear how to answer that neither. And it's very time consuming to experiment. So yeah, that's my best answer so far. Okay.
Okay. Any more? We could go to the next part. Okay, Julien, next part.
Okay, it's already 5:30. I mean 5:30, 4:30. Right? So we just have half an hour left.
Yeah, sorry.
Yeah. Okay. That's how it is. Okay. No, but I'm fine with that. Supervised modeling approaches. So second part. So I can go a bit faster. There, we've done this. Maybe interesting, yeah. So I showed you that figure from that review from the convolutional neural network. What people do now is, well, since a few years, is to try to go beyond short range dependencies, and that is motivated by the fact that in higher eukaryotes like humans, you have regulatory elements that can be quite far from the gene, from the promoter, so-called enhancers, that some have been reported up to 1 million days away from the promoter. It's actually not the majority. Most are in 10 kb or so, but still you have really enhancers that can be further away, and so one needs to capture that. And so methods for modeling this have been pioneered by Kelly, David Kelly, now at Calico, with this paper Basenji back in 2018, where he used for the first time dilated convolutional layers, which allows you to have an exponentially growing receptive field as the number of layers. And so this allowed him to have already a model that has a 32 kb receptive field, so capturing quite a bit of the number of enhancer promoter interactions we can see. We also use a relatively low resolution, 128 base pair resolution, but still was also modeling in a multitask fashion all the several 4,000 and more encode tracks. Okay, so that gave one of these first papers where we could really see this tracks predicted at that level of accuracy, as well as those predictions that are close to replication performance for many assays. Okay, and then another part that is in this kind of landmark paper is the interpretation. So what you see here is across the genome, the effect of single nucleotide variants on the predictions. And so that gives you these important scores here, which allows you to identify then regions of importance and particular motifs. And so later on, people, including especially Anshul Kundaji's lab, has developed methods to extract from these important scores motif a bit in a way that bioinformatics was doing before, by enrichment of k-mers or so. You get motifs that are extracted from those tracks, and we can recapitulate much of the position weight matrices that had been identified before, at least for humans. There's not so much that has been discovered in humans with these techniques. Actually, the lexicon was relatively complete, but you can recover them, and you can see also importance of their dependencies, which were not well captured so far. So far, so good? Okay. Good. And yeah, later on, Informer, which is perhaps still until this bozo is completely published, remains maybe the best model out there for these types of problems, called Informer from Giga Alsets, who has then moved to a transformer architecture. So using attention layers to model these interactions and go beyond technically, beyond what Basenji could do, and go up to 200 kb parameters. Yeah, but not 200 kb receptive field. Now Alex Carlos is a PhD student in my lab, so he looked a bit, assessed quite systematically those tools. So what he's done is took a large set of assays that had been published, where people have looked into with synthetic biology into enhancer promoter relationships by performing either mutations in the genome or activating here with CRISPR some enhancers and seeing the effect on the gene. And what is reported in those assays is that there is actually way more long-range interaction than what Informer is predicting. So there is a dependency of how important an enhancer is, or regulatory element is with respect to a promoter. That is a typical power law decay. But the power law decay that you see here in blue, that is the one from the informer, goes much deeper than what those assays are reporting that you see here in red. There might be a bit of bias. The assays tend to report things that are positive, perhaps. But still, we have not only just this data, also data coming from genetic associations from EQTL that shows that these models, despite looking at a very large receptive field, still don't get which enhancers are going to which promoters. And so there is still room for improvement there in cracking that layers of regulation. Okay, then that allows me to go to self-supervised modeling, since I skipped quite a bit. Is that fine? Are you still okay?
Yeah, it's good.
Okay, so now the next things, yeah, the first slides are published, and then maybe I still have time to go to some unpublished stuff. Good, so back to a few motivation slides. So I love that many of us love that quote from Eric Lander, who said that, or wrote that back, I guess in 2000 when the human genome was published. And he said, well, genome bought the book, hard to read. If you look at a genome, you have these huge sequences of ATCGs. And what we want to do, and all these supervised learning tasks are doing, they're predicting some measurement, encode, gene expression, whatever you want, from the sequence, right? So you make this big jump here, predict Y from X. But perhaps, and that's the concept of representation learning that is quite developed now in machine learning, perhaps with better representation of the inputs, with, for example, self-supervised approaches, we may have representations that are then useful and make the training of the supervised model more robust, require less data than doing the full jump from the raw sequence all the way to Y. And I made this argument already, but right, again, selection is happening. We have a lot of species, and so we have many genomes out there which are enriched for functional element, and learning how to reconstruct genomes with mass language modeling is a good way to have good representations of sequence. At least that's the motivation of our lab and many others that are developing and investigating this approach. There's nothing new conceptually. I mean, conceptually, there is a bit of novelty, of course, but this is something that people have argued for a while ago. That's called comparative genomics. Yeah, so comparative genomics is this idea that if I compare genomes and I find a common point, probably I find also things that are functional, and that's a way to then study those elements. But the big thing is more a bit algorithmic here. Except everything that I'm telling, that we have way more data to do this today. What people have done in comparative genomics largely is based on a workhorse algorithm that is the sequence alignment. And the idea behind the sequence alignment is that much of what let's put it like this. In many situations, you have an ancestor sequence from an ancestor species that has then been inherited and evolved across all our species. But that is a common ancestor to sequences that we see now in the genomes of those species. And aligning the current species gave us a bit of an idea of what this ancestor sequence was. It is, however, this approach has been powerful. So you can do this exactly in this setting that I'm telling about, which is looking at homologous sequences that indeed descend from a common ancestor. But you can also do more generally by aligning sequences with some anchor point. I'm showing you here another example of using this concept that is not necessarily limited to homologous sequence. You can take, and this is really a textbook thing, you can take sequences of protein coding genes and align them by the start codon. If you do that, what you see around the start codon across various protein coding genes is some sequence preferences. In particular, this third nucleotide before the start codon is usually favored to be an A. And it turns out that if you do an actual translation assay and have varied the value here, all the rest being equal, you will see that if you have an A, the translation is more efficient. It helps the ribosome to assemble, to recognize that start codon. So things that are favored can be beneficial. Things that are disfavored tend to be detrimental. Here you have, yeah, okay. But that requires this alignment to be done. And this is not always the case, that you can nicely align sequences, even though sequences may relate functionally. This is well known. You see this in any part of the literature, especially when you go to non-coding sequences. I'm showing you one example here from a recent paper, but just to illustrate that you grab that. What you see here at the top, S-Cerevisiae, that's Baker's yeast, and we're looking at one gene called CBP3. And the 0 corresponds to the stop codon of that protein coding gene. And here you look at 200 nucleotides that are 3' of the stop codon. So we are in the untranslated region of the CBP3. We know this is the untranslated region for Cerevisiae because they are good annotations, but it is whatever sequences we have 3' of the stop codon of that gene. I then go into Paradoxus and Baianus, etc., all the way to Pombe. This gene has an homologue. You can see this from the protein coding sequence. You can grab this, and then you can extract again the 200 nucleotide. We're not aligning. It's really the raw 200 nucleotide for all those sequences. Then what we do is we take BLAST, which is the typical sequence aligner, and we, with default parameters, we align the sequence, and we ask, is the Cerevisiae, my starting species here, is this region aligning to that region of Paradoxus, of Baianus, etc., all the way to Pombe. And we can align with BLAST default parameters or within the so-called genus. So as long as it's a Saccharomyces, it starts with an S, this is okay. And then Pombe is actually not from the genus. And then for the other species, we lose. We're not able to align. You may play with the parameters, but eventually it's just, you lose the alignment. And one reason for that is probably that the untranslated regions are quite flexible. The order of the elements is a bit flexible. It's not as constrained as a protein sequence. So alignment works well for protein coding sequences. In non-coding sequences, it doesn't go as deep in terms of evolution. However, what you see here with this red box is a motif for an RNA binding protein called PAF3 that is important to determine the half-life of this RNA. And PAF3 happened to be a protein that is very ancient, and you find that RNA binding protein in all the species. You even have an homologous in human. Moreover, if you take this motif and you look for it, you will find it also in essentially all those UTRs. You always find it significant in this species, indicating that perhaps actually the binding to RBP to PAF3 matters for all this for this gene across all the species. And in fact, there's even experimental evidence for a species that is quite, I think, 400 million years evolutionary distance away from Cerevisiae for a functional PAF3 motif. So this suggests that we do have an element that is conserved. It's the same. One should statistically, I mean, with some algorithm, be able to find that if you know what you look for, perhaps. But the alignment is not the right way to grab it. You lose the position, right, of that. And that can be rationalized because UTRs have variable lengths, because these motifs may not have to function exactly at the same position. So you can still have the function without necessarily having an alignment that is traceable. Okay, And that is true for UTRs. It is also true for enhancers, for many sequences, although as soon as you move away from coding sequences, you can't go as far as, even though, as far in evolution, even though the function may be conserved quite far off. And therefore, we and others have embarked into this approach, which is let's use alignment-free approach and use mass language modeling. So that's a work from Alexander, Johannes, and Dennis that we published just last month, I think, in Genome Biology. And what we've done there is we took a transformer that takes as input a piece of DNA, and it is trained, as very classically in a mass language modeling approach. You mask 15% of the nucleotide, and you train your model to recover this nucleotide just by a cross-entropy loss. Our innovation here is to say we train this across species that are very distant from each other. So we provide to the model also another token for the species, and it learns how to represent that token so that the reconstruction is good across species. Okay, and then I will show you about later on that we use that as a foundational model for some downstream tasks. So far, so good?
Yeah.
Okay, good. So the core hypothesis behind this, again and again, I'm telling this, but indirectly, this model that learns regularities in those genomes should capture functional elements of the genome. It is not true that everything that those models can recover is functional. Yeah, you can have also things that are easy to guess but are not so important, or they may not be important for any tasks, but hopefully things that are important are part of what the things can capture. Okay, good. We train our model across more than 800 species. We downloaded the entire assembled genomes from Ensemble for the fungi, so fungal genomes, and that spans over 500 million years of evolution. Among the fungi, one species was of particular interest for us, and that is Saccharomyces cerevisiae. It's the Becker yeast because it's a model organism that is extremely used in biology. There's a lot of label data there, so we can do a lot of evaluation. In the context of this paper, we took a very, let's say, skeptical point of view with respect to DNA language models. So we pushed it a bit far in the sense that we removed Saccharomyces cerevisiae from the training data set. We also removed the entire genus. I showed you this is where sequence align well, so that the model is not really learning by heart from the other species, this genome, that we have a bit of distance. And we tested then how this is performing and predicting those guys. So in order to reconstruct the sequence of cerevisiae, we will take the closest species evolutionary from Saccharomyces cerevisiae as a fake species token just to proceed. We are not claiming that this is how those large language models should be learned in the future, but it's really to evaluate the power of large language modeling that we have done this relatively strict approach in the context of this paper. Okay, we train two models. One model is upstream of the coding sequence. So the coding sequence can be easily mapped with classical bioinformatics tools across those species. So Ensemble provides you with an annotation of the coding sequences. Ensemble does not provide you, however, with an annotation of untranslated sequences. Yeah, so we don't know whether transcripts are starting or ending. Actually, we know this only for very few species for which this data are there. For most of them, it's unclear. We do have, however, bioinformatic annotations of the protein sequence. So we anchor our model exactly upstream of the start codon and downstream of the stop codon. This was on purpose because we believe that, or we know that there is quite dense information close to the coding sequence. And so that's what we aim to recover. At the same time, it's the non-coding part, so it's still also interesting for regulatory purposes, for regulatory modeling. This upstream model is fixed distance of 1,000 nucleotide. It typically covers the 5' UTR, which are shorter than this. It also typically includes the promoter of those genes in those species. And often it even goes up to the upstream genes. Same for the downstream model. It's a bit shorter because here we want to simply cover the typical 3' UTR, and there's a bit of sequence, a bit heterogenic downstream. This sees 10 billion bases, so we can have highly parameterized models. We use a transformer with about 100 million parameters. That's still not as much as we have data. Yeah. Okay. So we trained that by mass language modeling, very standard, nothing fancy here. In the paper, we've looked at tokens that are overlapping 6 mirrors, but now we've seen that we can actually have tokens which are just the single nucleotide, and all the results are the same. So you can think of what I'm showing you with a transformer training on single nucleotide tokens. The first thing you see here is what is your probability of a nucleotide given this context, or these windows I just showed you before. I'm showing this here with a relative information content relative to the ground, to the general probabilities of each of the nucleotides. And what you see is that in this region is upstream of the start codon of that protein here, RPL36A. Upstream of that start codon, we have a bit of the untranslated 5' UTR region, and then we have the promoter. This is a ribosome genes, and there are motifs showing up here that are well known. RAP1, for example, which is a motif of a transcription factor involved in ribosome regulation. The sequence is large, and it goes into the untranslated region of another gene that is located 5' of our anchor gene. This is YMR193W. And again, you see regions that are better reconstructed than others. And these regions correspond to motifs that are important for 3' UTRs. One is the PAF3 motif. I mentioned to you this PAF3 motif a bit earlier with my motivation example on alignment. And the other one is a motif that is involved in the efficiency for recognizing the polyacide. Okay. So this shows that just this model does the job, at least on this example. It does reconstruct. It is not the goal of this to reconstruct everything perfectly. That would be almost boring. That will not distinguish important from non-important things. It reconstructs better things that are more important. And you have to think that also in biology it's the same, right? There are many places in the genome where mutations can happen and it doesn't matter. So it's also fine if the model is not able to reconstruct everything perfectly, because it would be probably an evidence of overfitting. Okay. We actually had some time to realize that. So students in my lab were really crying on this project at the beginning because they didn't see their loss changing much. And the global loss, it's driven by all those bases that you don't reconstruct so well. We're basically guessing just with simple dinucleotide frequencies also. It's just doing the job. And then there's a little bit in the loss, which is driven by those things that are actually functional. So you don't see much just looking at the loss. You get to look at things that actually are known to matter from orthogonal data to see the advantage of your model. And that's what we did. And I'm going to show you now things that I find a bit surprising how well this works, because it pushes us basically looking at things that are a bit beyond what we could do before. So as I mentioned also in my motivation, finding the lexicon, this is not too badly done. Finding the grammar, the context of where things work, that's where bioinformatics has been so far a bit more limited. So here, well, I'm looking at this is a true motif. That's this PAF3 motif. And in this cartoon, I'm showing basically the motivation of the data I will show you. I'm asking, how is the model performing when it's asked to reconstruct a particular sequence which matches the consensus motif for an RNA binding protein, when the position is actually within an RNA, suggesting there's probably a true functional site for this RNA binding protein to act. And when you find the very same sequence, and those occur, there can be sequences like this in the genome, which are just not happening to be an RNA, and therefore don't have, or less likely to have a function. So we do that. We go throughout the genome. We scan with the PWM, the position weight matrix, the genomes in those regions. Much of those hits are within the RNAs, but there are still some which are outside the RNA. And then on the y-axis here, we ask, what's the log likelihood of reconstructing, in average, those bases? And you see that those that are actually within the RNA are better reconstructed. And what you see on the right-hand side is that this is true for the species LM. You do actually quite a good job with the agnostic LM, perhaps because PAF3 is present in all those species, so it's not so important to be species-specific in that very instance. But if you use alignment-based conservation, and this is available for Cerevisiae within this genus, then you don't make much of a difference. So those things align also, because maybe you don't see so far in evolution with alignment-based conservation. Then you can see with a DNA language model that is able to train with all the data. So that's quite remarkable. That means the thing has learned not just the motif. Underlying, there's this context of, are you still in the RNA or are you downstream of the RNA? So where the poly A site is. It has to be trained because this alignment that I'm doing here, this alignment in the sense of putting all the data together, aggregating across genes, I do this by centering all the data in that 0 here, which is the 3' UTR end, which is experimentally measured in Cerevisiae, but the model didn't have access to that. It had access to the stop codon. These UTR varies from gene to genes. We are aligning them. The model didn't have access to this information. The model was not trained on the Cerevisiae genome, and still it captures the context where this thing is actually functional. So I found this very exciting, and we have other examples of these also in the promoter. This type of models can be helpful also for downstream tasks, and that's the motivation of many papers. You take this reconstruction as a pretext task. That's not what you're really excited by. But once you have trained that, you probably have a model or even just a representation of the sequence that can be used for downstream tasks. And in this paper, where we're really after just evaluating the quality of the model and not necessarily focusing on the downstream tasks per se, what we've done is we really froze everything. We just extracted the embeddings from the 4 last layers, and we've done just a ridge regression, just a regularized linear regression, just to assess how good this is. And it was surprisingly good. So here what I'm showing you is maybe the most conceptually difficult task for these models, because we're asking the model to generalize on non-endogenous sequence. Yeah, the risk of these large language models is they learn correlations, they learn correlated features, and these correlated features are predictive. But as soon as you put things that are not natural, like as soon as you mutate the sequence, they may fail. So we train them on exactly data like this, which are these kind of synthetic biology data sets that I told about in my motivation, or in my earlier slides. These are data from Eran Segal lab. They have identified 200 genes for which they took the untranslated 3' region. And then for each UTR, they went in a tiling fashion by regions of 10 base pair, and on each construct, one of these 10 base pair window sees all its nucleotide randomly reshuffled. So you get the same GC content, the same nucleotide content, but you differ from the endogenous sequence with one of these windows being permuted randomly. Okay, and this is for 200 times all these time windows, so we have about 2,000 or so sequences. And we can train a model in a cross-validation fashion. We were removing the entire genes, right? We re-stratify by gene, and we were able to remarkably well predict then on held-out data the expression level of those reporters, capturing really the some of this half-life or poly A site definition code that is in the 3' UTR. So the left-hand side is a good example where we have very perfect, almost agreement with the data, and on the right-hand side you see the global trend. So that's nice. That shows that we can really get state-of-the-art models. So this was better than the model that the original authors did, but 10 years ago, right? We just came years or so. And then we've done a bunch of other tasks which are more based on standard gene expression, not all based on MPRAs. And there we do also always with a simple ridge regression on these layers better than state-of-the-art models. So that was quite exciting for us. And so as a conclusion for this part, we could show that DNA language model captures conservation without alignment, that having a species-aware model allows you to capture evolution of the regulatory code. I didn't show you all the results on that, but you can check in the paper. There are nice stories where we see motifs that we find in some species and not others, and the model reconstructs them in the right context. And then they can function and operate as foundation models, and for this, they can provide effective representations for gene expression traits. Okay, so far so good. And what time is it? Yeah, but it's out of time. Okay, so I think that's good.
Thank you so much, Julien. Sorry about the late start. Let's give Julien one more round of applause.